Hello, and welcome to this Pharmaron podcast, part of our DMPK Inside podcast series. My name is Chunyan Han, and I'm the Executive Director of In Vitro Academy, DMPK at Pharmaron. Today, I'm thrilled to be joined by Professor Amin Rostami, a leading figure in the field of PBPK and QSP. Professor Rostami is a professor of systems pharmacology and director of the Center for Applied Pharmacokinetic Research at the University of Manchester, as well as the CSO and senior vice president of R&D at Cetera. Welcome, Professor Rostami.
Thank you very much, Chunyan.
Thank you for joining us today, especially as we celebrate your recent 60th birthday. Your contributions to the field of pharmacokinetics and pharmacology have been truly remarkable. In your current position at the University of Manchester and Cetera, your work has significantly shaped the landscape of drug development. We're excited to learn more about your journey and insights. So to begin with, can you please let me know what factors initially sparkled your interest in PBPK and inspired you to pursue a career in this field?
First of all, it's a great pleasure to be with you and Pharmaron colleagues, for creating this particular podcast. And going back to your question, by background, I am a pharmacist, and I started actually as a lecturer in pharmacokinetics and did my PhD in clinical pharmacology and clinical pharmacokinetics. So my start of the PBPK was some kind of, I would say, coincidence that, I took part in an interview for a job before getting my permanent position as lecturer in pharmacokinetics in University of Sheffield. And as part of that, when the interviewers were asking me about different angles of my work, they basically praised everything I had done. But at the end, they said this particular post, we would like the person to have some background also in the PBPK, which I had heard about, but I had not worked on PBPK. So anyway, I got the the permanent position in University of Sheffield and didn't go to that particular post, but it intrigued me to look into what actually PBPK details are. And at the time, we are talking about mid-90s, this was something that mainly was, I would say, led by, pushed by our colleagues in the environmental tox area. So the more that I looked into the aspects of the physiologically based pharmacokinetic, let's say, research and advantages that it can bring up, the more I became concerned that there are many elements of the in vitro in vivo extrapolation that they are not adequately used within the frame of the PBPK. So that was the start of basically me getting into the PBPK, and with the background that I had in pharmacogenetics, pharmaceutics, and clinical pharmacology, I could bring many of those elements, starting with the drug metabolism and drug-drug interactions, as you know, and the creation of the SIMCIP consortium, into the fold of what people nowadays call new PBPK or PBPK IVE, in the form of the tools we generated and the philosophy of the work, which has now become ingrained in majority of the leading pharma.
Okay, great. You just mentioned about the SIMCIP. I think your career in PBPK has been closely associated with the development of a SIMCIP simulator. And can you please share more about the founding of SIMCIP Limited and development of the software? About how did the idea come about and what was the initial challenge in its development?
Yes, the background that I gave you with regard to me entering into the PBPK culminated in the form of one of the undergraduate studies that was done to work out what we basically in our field call stochastic prediction of the range of the drug-drug interaction between methadone and ritonavir. The student who was doing that, Adam Tooley, was presenting this work in one of the meetings that majority of the pharma-related drug metabolism people they actually attend. This is no longer continued. It's called DMG meeting, not DMDG, but DMG meeting. And when he presented the work, several of the industrial colleagues came and asked whether what he has done in the form of a MATCAD model come up with simpler interface in a way the colleagues that they have got in these pharmaceutical companies can make regular use of the approach that he has taken for that particular pair for other drugs as well. So the starting point, in fact, one of the people, who were involved at the time, from, the old Pfizer, uh, is still in, I think in the field and works, in fact, farmer on, Barry Jones. Um, and several others, they decided that they are coming together as a pre-competitive project to take lead in the conversion of this scientific piece of research into a tool that is going to be used by their mm, teams within their pharmaceutical companies. So that was the start of the, I would say, pre-competitive collaboration as a project in 99. Uh, University of Sheffield, I think in 2 years after that, they decided that this conversion can happen through a spin-off company of university, and that was the start of SimSip project being transferred into a spin-off company of SimSip Limited by University of Sheffield, and eventually this particular entity, as many of you will know, in 2012 became part of Certara. So the history of the SimSip as a platform for PBPK modeling, and particularly the population variability, um, started as a small project by 5 companies on the back of DMG discussions over dinner, but then rolled over to a spin-off company that later on went well beyond drug-drug interaction and many other areas, disease populations, and the oral absorption of pharmaceutics, as well as other routes and so on.
So have it been very smoothly since it had been created?
No, as with many other big projects, there are many, many challenges involved. But I would say that on the positive side, this has been one of the longest running consortia ever that has been focusing on a certain area. As you know, and many others may not know, that SimSip consortium members gather every year and they decide on the direction of the additions that they are going to bring into the platform. So this has been a positive element, but the challenges, of course, have been that many different people they have got different priorities, and settling on these they are challenging. And also, as the areas that the platform has been covering has moved, for instance, from just small molecules and DDI to many other areas, including biologics and so on, the expertise that were required has become much more diverse, and getting all these people talking the same language, understanding each other, and having a platform that is I would say compatible with all different elements of it has not been an easy task. But I'm glad that over the last, this is getting into 25th year this year of the SimSip consortium, that over the 5 years of the consortium, we have largely overcome such challenges.
Congratulations for the over 20 years for the same SIP consortium. I think you just mentioned changes in PBPK. So how has the field of PBPK evolved over the decades of your career, and what have been some key milestones?
I would start by going back, to where we started. The SimSip platform everybody is familiar. It started with the drug-drug interaction part of it, and later on went to many other areas that it covers. So I will start by saying that the in vitro elements that people were doing, perhaps, they have not majorly changed. Of course, we have got many new techniques and new systems, etc., but the principles, they have remained more or less the same. But the philosophy of using such in vitro systems over the last 2 decades massively has changed, because originally many of these in vitro systems, they were used as a way of ranking different candidates that people had in their, let's say, lead generation and so on, and bring up with lead and the backups and so on. And very rarely at the time, people were thinking of quantitative extrapolation to understand how those in vitro information, the numbers that they are getting, can be translated into something that may address some of the clinical questions later on, and also fill up the gaps of the clinical studies that people are not easily going to be able to do. And this has been, I would say, the biggest actually change, to use the in vitro systems in a quantitative fashion, using in vitro in vivo extrapolation, using appropriate extrapolation factors, and using appropriate in vitro systems that they are representative of, the situation, and extrapolate those using the system information on these tissues in different populations, health and disease, or, let's say geriatric versus pediatric versus adult population and so on, or different ethnic groups, and make use of the same in vitro information that is against the fundamental elements like enzymes and transporters, but extrapolate to many different populations. So that has been, I would say, the biggest change, to make better use of in vitro data in translating them quantitatively into what will happen, and address some of the clinical studies that we no longer need to do, as well as actually doing virtual clinical studies in areas that nobody, easily could have done such clinical studies, yet the information are important when the drug comes to the market with regard to handling them.
Okay, I think your answer is kind of related to the topic in your recent talk at the Pharma Symposium, and you mentioned about the necessity of a high quality in vitro data and its rigorous analysis using tools like SimSip in vitro data analysis toolkit for effective data interpretation. So, how crucial are those detailed mechanistic insights provided by the complex model compared to the practical benefit of a simple model? Also, to what extent does the regulatory acceptance influence the decision between using a complex versus a simple model?
That certainly has been a big element of changes of the attitude towards in vitro data, because once these are accepted, in some cases in lieu of clinical studies, the burden of the quality obviously increases, and ability to generate them in a way that they are against certain references and standards becomes much more important. I think many people have seen the sometimes discrepancy between the results that, one lab creates versus another lab, and this was early on was a big issue for many people, which reduced their trust in some of the in vitro data. However, these days, by application of certain standards, that is actually been removed. But the other angle of it is that we no longer rely on these systems as a yes or no answer, but we are taking quantitative values from them and extrapolate on the basis of the content of these in vitro systems. For instance, the level of enzymes that the systems contain, or the level of transporters, and so on. So we can take these numbers, express them per unit of each of these relevant proteins, and then take those numbers into relevant models. And as a result of that, ability to analyze in vitro system data has become a big part of this IVIV that I already explained. If you are using in vitro systems in the very early stages of discovery, you may not need that, because the only answer that you are basically asking is still in the form of yes or no, below or above certain limit, and then you select and go ahead. However, when you are at the later stage, you want actually to know meaning of each of these in conjunction with all the other values of the in vitro data for other aspects of the drug kinetics as well as dynamics that you have got. And that is the time that the complexity of analyzing this in vitro studies become basically part of the game and part of the play. We noticed in almost, I would say 15 years ago, that despite that this is picking up, with regard to the acceptance, etc., but there are many actually big pharmaceutical companies who are not making the maximal use of their in vitro system. And we realized that there are not many people in the DMPK groups that they, by nature of, sort of the job that they are doing, which is mostly lab based, they were not actually engaged with many, let's say, modeling exercises. And we started to work on not only actually expanding on the models that they can use and apply to these conditions, but also come up with interfaces similar to SimSip, for the PBPK, come up with tools that they know what the structure of these in vitro systems are, what data goes in, what data comes out, and analyze them. And therefore, these systems will contain, with the models that they are associated with them, information on the nature of that in vitro system. For instance, what the abundance are, what the surface area of the wells, if you are using, I don't know, 96, sort of well, plate, and so on. And if they have got, three different phases, the donor phase, the cell lines, and, the receiver side, sort of, and so on. So you analyze the concentrations in all those, with whatever left at the end and recoverable, etc., and plug into a model. And therefore you are not talking about, for instance, for those people who are familiar with the permeability error, you are not talking about anymore just the A to B, B to A ratio, which may give you something at a given concentration, whether the transporters are involved or not, or everything is passive, but it is not going to help you to understand how the saturation and nonlinearity of the dose during absorption or later on during metabolism, etc., with, and if the transporters are involved, is going to play out. Because for that, you need to actually to characterize the affinities against those transporters and the maximum velocity that you are going to see against the concentration of such transporters in the target tissue. Therefore, the SIVA that you mentioned, SimSip in vitro data analysis tool, that was in fact a project, a grant that was offered by a UK government to the SimSip team to create something that help accelerate, facilitate the usage of the modeling when it comes to in vitro lab studies, with a view that the output can be then used into PBPK models.
Okay, sounds interesting. And I think in the Pharma Symposium, you also discussed about the concept of the model master file using acetinib as an example. So could you please elaborate on this concept? And how early in drug discovery and development do you think we should start to build the model master file? And do you see any difference in the strategy between biotech companies and pharmaceutical companies regarding building the model master file in their PBPK strategy?
Thank you for this question, because this is basically the direction that the modeling in the regulatory agencies more or less is moving towards. For those people who are not familiar with the concept, earlier this year we had the first workshop specifically actually dedicated to model master file. This was earlier in May and organized by the University of Baltimore and FDA jointly in the center that they basically focus on innovation and research excellence in the regulatory area, CERSI. So people can go and look at the presentations and discussions and debates that happened there. I believe it was 2nd and 3rd of May this year. Um, the idea is similar to having a drug master file. So a drug master file, those people who have been involved in any regulatory submission, contains all the information that you have generated with regard to the drug when you are submitting to FDA, and all the time it is uh, complemented by any additional information that you are bringing and putting into that file. So the idea of the model master file is almost in the same line. It is suggesting that many aspects of the modeling for a drug that's coming to market that can be amalgamated in the form of a master model, and anything else can be added to that model going forward. And the example that you mentioned, it is not unique example. The one that you mentioned is related to what Novartis had done with one of their compounds, but there are similar examples in the case of AbbVie, as well as other examples in the case of Eli Lilly and Pfizer and so on. So the way that it operates is that you build your PBPK model, let's say once the drug is selected and going through the drug development. So we are not talking about the discovery stage when the models are, let's say, many aspects of it, they are uncertain and so on, but you have got some certainty over some elements of the model going forward. But from that moment onward, whatever additional information you are bringing, that goes on top of what you have created and gives answer to additional questions beyond the original purpose of the model. So when people talk about fit for purpose model, we are not talking about here fit for purpose model. What I will call about these particular models, they are fit for purpose but framed for future. They have got this openness element about them that enables them to be expanded and have branches going forward. Therefore, while the basis of the PBPK is the same, there are elements of it that they are addressing additional information that you did not originally meant to actually address by the first PBPK model. So you may start a PBPK model to develop it for addressing drug-drug interaction. But later on, you may decide to address the issue of adult back extrapolation to pediatric dosing. And this basically the use of the model master file. Even you originally did it for DDI, now you are extending it to the pediatric application. Then you may want to use that again upon some applications and verifications. You may want to use it for ethnic differences, for genetic differences that they are part of the ethnic differences, and so on. Therefore, the model master file during the life cycle of the drug is used and reused and reused and reused. And therefore, you can see, for instance, when it comes to the pharmaceutics application side of the thing, which, the example of AbbVie particularly has been focusing on, it may address, for instance, a change of the pharmaceutical firm that was making the tablet and so on, or some of the ingredients that or the particle size and so on. So you are using the same original, you are not building a new PBPK model, you are using the same PBPK model because many aspects of it is already established, and you have got adequate, I would say, confidence in those elements. And also, you are not basically every time changing the model, with something that, was working before and now it's not working anymore. This gives, sort of the, I would say, higher confidence that in many different conditions, this particular model is going to perform. And you are gathering more and more evidence for that. Therefore, the model master file is an attempt to organize, centralize, some modeling effort that eventually they may go even beyond, the fate of the drug and PBPK, and connect, for instance, to quantitative systems pharmacology models, and address also the dynamic elements of the effect and so on.
So, do you see any difference in strategies between biotech companies and the large pharmaceutical companies?
Yes, that element, I forgot, to actually to bring up. While the model master file, can be applied both with the big pharma, with those examples that we mentioned, and also with the, I would say, smaller biotech. The difference is that in the majority of the big pharma, they do have all the expertise that they can handle different aspects of these model master files. However, with the biotech, because of the nature of, they are small and nimble, and they can move faster, but at the same time they can't address every element of these themselves. So typically they will be using partially the creation of such model master files themselves, but in many cases they will be working with the outside collaborators, CROs, CROs in the space of in vitro, as well as CROs in the space of the modeling, to create such models. And many of these companies, because they eventually end up by selling, their assets to bigger pharma for the completion of the development, this becomes handy when they are going through due diligence, because many big pharma, they are familiar with these concepts, and they take, I would say, keen eye on understanding how these, let's say, virtual clinical studies that you are running on the back of these model master files are performing, rather than just relying on certain, I would say, raw data on numbers. And therefore, while model master file is used both in the big pharma and the small pharma, but in the small pharma and the biotech, you may consider them as a, let's say, element of the due diligence that they will go at some stage when it comes to selling their assets to some bigger pharma.
Okay, thank you for sharing insights into your research. I'd like to shift our focus a bit and talk about your current role at the university. So, as a director of Center for Applied Pharmacokinetic Research at the University of Manchester, what are some of the key research areas your team is currently focusing on?
I have to admit that I have been in this unique position now for roughly 25 years, of having 50% of my time dedicated to the business of Certara, and before that, SimSip side of the consortium and development of the tools and platforms. But the other side, I have remained in the academia as a professor of systems pharmacology, and that has given me, I would say, the unique opportunity to see all the gaps that we are having when we are trying to create these platforms and models for different purposes, and do my best effort via different grants, and sometimes by collaboration with the pharma again, to create the numbers information that they are filling that gap. So when it comes to what we do in CAPCA, I would say that we can address them in three different levels. A big chunk of what we do, they are related to creation of system information. And these systems information that we are talking about could come in the form of abundance of transporters, abundance of metabolizing enzymes and receptors, and all other relevant proteins in different tissues, in health and disease, and in different sets of population. And these feed into all those platform, let's say, and the clinical virtual studies that they are going to replace clinical studies. Therefore, understanding what is the difference of abundance of these targets in, let's say, for instance, if you are talking about skin, uh, between Afro-American versus, let's say, Caucasian, uh, groups is in some cases might be important. Or if you want, to go from, for instance, from Caucasian to Japanese with regard to some of the transporters in kidney and liver, how the abundance, is going to be different, that is of our concern. Same with regard to the adult versus pediatric. Same with the disease. For instance, we have done some work in biliary atresia in these young neonates, versus, sort of the people who haven't got that disease for the pediatric as well as in adult. So one part is basically generating all such data, systems data. Major part of it is proteomics, but it's not just proteomics. We have got other elements that we have worked on. The second element is creation of the models that we have not played with, we have not tested them. So whenever the existing models, they are not capable of addressing all our needs, we will be working on new models to be generated. For instance, at the moment, we are working on PBPK models of direct nose to brain access of drugs getting into the, let's say, our brain and having their effects. But part of that, of course, involves the mucociliary, clearance going into GI tract and so on. And we are building the models and trying to test them. But then the third element of what we do is, as part of this translational modeling that we are putting to such new models, we have to also verify the models. And for that, we may get engaged with clinical studies. And therefore, for that particular case that I mentioned in direct nose to brain, we have got studies that they are involving with collaborators we have got in University of Barcelona, that they are looking into IV as well as intranasal administration, so we can use the deconvolution and understand how the absorption is happening and the kinetics of it. Therefore, there are different layers of what happens in Center for Applied Pharmacokinetic Research, but I would say the bottom line is that the information that we get from setups such as SimSip Consortium and the gaps that they are existing in the PBPK models, and also going forward with the QSP in the similar vein, they help a lot to define what kind of activities we will be doing in CAPCA.
Okay, I think my next question, I plan to ask how you balance your academic role as a university and with your industry role, etc. But I think you have already combined them together, maybe?
I would say with lots of difficulty. The biggest challenge is that, both sides, they are assuming that you are 100%, sort of working for them. And therefore it has been difficult, but it had its rewards, as I explained to you as well. But it has been challenging. So at the same time that, I may recommend to people, if they can come up with such position, to take it, I have to also warn them that it is not an easy task.
So, how do you see as a future trend in the PBPK and the QSP, and how should researchers prepare for these changes?
I would say that the biggest thing that I can bring up in the, I would say, transition of the activities and mirroring them for the QSP is that they have the advantage of not making the same mistakes and going faster, knowing that what works and what doesn't work, because obviously the PBPK and in vitro in vivo extrapolation quantitatively started in the PBPK earlier. But because we know their roadmap, I think the advantage is that the QSP can take advantage and of all that what we said and go faster. On the other side, when we look at the QSP difficulties that they are associated with it, is that unlike the PBPK, it is not something that is common with every particular company that you are looking at. While the fate of the drugs, whether it is small molecule or large molecules, they have got many common threads that you can come up therefore with some position with regard to creating a system that addresses everyone's need. When we look at the QSP, it basically depends on which area you are looking at, whether it's in the neuro area, whether you are in the metabolic disease, whether you are in the oncology area, and if the oncology, it's immuno-oncology, if you are looking at other bits. Therefore, actually the task multiplies by the number of the disease area that you are looking at. In some of those areas, the systems information, call it systems biologies and systems, pharmacology and physiology, they are much better known than some other areas. But gathering that information into one place, it has shown to us in the PBPK, it adds much more. So the sum of the parts is much more than, the individual, sort of pieces put together. So in holistically, they offer something that we couldn't get just by adding the pieces together and assuming that they are going to be just added value. It is much more than that. It creates lots of synergy when you put them together. Therefore, I am very, very optimistic with the QSP that it will go much faster than what PBPK has done. And with the AI coming to the help with regard to gathering of the data, and in many cases also helping to put the models together, I think this is going to even, to go faster than just, having the roadmap. Not only we have got the roadmap, but we have got the tools that helps to go through that roadmap much faster.
Thank you. I think my last question is, what advice would you give to young scientists and researchers entering the field of PBPK?
My biggest advice would be that you should not be afraid of the modeling. If you are a lab-based person or clinical person, I think separating the modeling and thinking that is something that some clever people with lots of mathematics do in the corner, that we have passed. Modeling is now much more, I would say, has got a broader element. Of course, there are a group of people who are going to create and put those all the mathematical know-how and this know-how is go is going to be still with those people who have got the skills there. But I think the broader application of the modeling now comes with the people who are experts in their own field, and these field expertise help them to ask the right questions and then search for the right tools to answer those. So therefore, my recommendation would be to all those people who are working in as a non-modeler to get engaged with more modeling as much as possible. And I guess that Chunyan, in your own case, you have I would say enjoyed to some degree, even it was a tough sort of case, but the modeling that you did for the first time for this I will say local, I will say, bioequivalence in the Crohn disease in the GI tract, which nobody had done before, and how this actually the translation would be very different. This is a kind of application of the modeling that you may say that the mathematics of it was already there, the models were there, but nobody has been thinking of how to apply these for a practical question that everybody had and nobody had any answer for. And if you want to answer those questions, which you have done with your virtual studies, you may need, God knows how many clinical studies with how many different drugs, which basically makes them practically impossible.
Yeah, I think making our PBPK modeling, applying them to the real world situation, making them more meaningful.
Thank you, Professor Rostami, for sharing your insights and experiences with us. Your contributions to the field are truly inspiring, and we look forward to seeing how your work continues to shape the future of drug development. Thank you all for listening to this episode of Pharmaron's DMPK Insight series. We'd like to remind you that our DMPK webinar series is also available on demand, covering a variety of key questions related to DMPK sciences in drug discovery and development. Stay tuned for more podcasts in our Pharmaron DMPK Insight series. Thank you.
Bye, everyone. Thanks for the opportunity.